Hi, everybody. Thank you all for joining. I hope that you're all introducing yourself on chat, letting us know where you're from, what you're up to. It's really great seeing you all here today, and thank you so much for joining. Team ICAO and I are super excited about today's webinar, and we hope you're all ready for a mind-blowing journey into the lives of the little guys beneath our feet. And as we get ready to start, please go ahead, use the chat, as I've said. Let us know where you are, what you're up to. It's super to see so many people from all over the world. We've had a very, very large attendee booking for this event, right up to 300 plus individuals. So our Zoom is going to fill up, and anybody else will just have to come in on the YouTube live, and hopefully they'll hear us only once. Okay. So the ICAO webinars are brought to you by Team ICAO in Nairobi. We're a passionate company that optimizes smallholder farming practices and systems in Africa through our ICAO regenerative agricultural e-extension platforms and ecosystem. At the end of today's presentation, we'll have a question and answer session. So please forward any questions you have for our speaker via chat. We're going to be quite a big group, a huge, fabulous group. And unfortunately, as you can imagine, we're not going to have time to answer all your questions, but we shall endeavor to get through as many as we can in the time that we have. And on that note, I'm really honored to introduce you to Wesley Saul, who's joining us from South Africa to help us with the Q&A across Zoom and YouTube. Let's all say a big hello to Leslie. You can do it with your reaction button by sending heart or thumbs up or whatever it is that you can do. Wesley joining us shows us that this is actually truly a really continental collaboration in bringing some of the greatest minds in the areas that we're looking at, regen ag, into our domain. So Wesley, over to you.
Thanks very much, Sue. I'm very excited to be here today to listen to James. Firstly, my role here is just to answer and manage the questions. So I'll be saving any questions that kind of pop up. If you want any clarification, James has indicated that please just say, can you sort of give some more information on that? And especially if one or two people will say that, then I'll know it's something that needs clarification. I can just bring that up directly. That is the one advantage we have here with the live chat. I'm in Cape Town and very excited to be part of this among all the other Soil Food Web people. I can see there's a lot of us, and yeah, I don't want to waste any of James's time and get straight over to him. So thank you very much, Sue.
Thank you, Wes. I really, really love the continental collaboration. And finally, yes, you're right. Let's get started. It is such a great pleasure and an honor to have Professor James White join us today in what is by far our largest audience to date. His subject matter is set to change the way we think about agriculture, literally from the ground up. Professor White is in the Department of Plant Biology at Rutgers University, where he teaches courses that pertain to the biology and ecology of fungi. His research involves evaluation of the function of the plant biome, non-pathogenic microbiomes that colonize healthy plants. I'm not going to say much more. I'm going to leave it to you, James. So over to you and welcome.
Thank you so much, Sue. I really appreciate you guys inviting me to do this presentation. It's a big opportunity for me to speak to so many people in Africa. So I really appreciate the opportunity to talk to your audience. I'm going to share my slides. And so what am I going to talk about? I'm talking about plant microbiology and really how plants grow using microbes, using soil microbes, fungi and bacteria. And in specific, some of the processes that I'm going to talk about are these processes: rhizophagy, really rhizo for root, phagy for eating. This is how plants get nutrients out of soil microbiology, out of their microbes in soil. This is why we want rich soils. I'm going to talk about seeds and how seeds really are vectors of these microbes, that they are like a Noah's Ark, really, for the plant, in that they contain the embryo, the seed, the baby plant, and then then they also contain all the microbes the plant needs. That's the healthy seed, right? Healthy seeds grown in healthy under microbial rich conditions and microbial soils will have microbes with them. They're really important for survival of the plant. Also, I'm going to touch on the aspects of food security, and that is the effect on microbes microbial effect on plants in making plants more hardier and more resistant to extreme climate perturbations and environmental stresses. So really, microbes in plants are extremely important for all of these for all of these areas, and for our food safety, our food security, the fact that we're going to have, we could have environmental catastrophes, but endophytes can make things much better, and I'll explain how that works. So topics that I'm going to cover, I'm going to contrast biological agriculture versus conventional agriculture. Explain those. Biological, I'm referring to growing plants with microbiology. I'm going to touch on what are endophytes. These are microbes that go into plants. So microbes absorb them into plants. They're non-pathogenic. Internal phyte for plant. And we'll talk about that. The rhizophagy cycle now, and that is the nutritional, the rhizo root phagy eating cycle. The roots are absorbing microbes to get nutrients out of them. I'm going to talk about how microbes adapt plants to their environment and to stresses in a process called habitat adaptive symbiosis. And this is how microbes really determine the hardiness of plants and the ability of those plants to survive harsh environments, harsh conditions. And then I'm going to talk about, finally, how bacteria really are the immune system of the plant, or these plant microbes, soil microbes and plant microbes are the immune system, or a good part of the plant's immune system. And they suppress disease, and I'll explain that. And so those are the main topics. So just to contrast conventional agriculture, this is really chemical agriculture. Okay, this is chemical agriculture. This is what we do. We basically, for the most part, in industrial agriculture, we manage crops using chemistry, and we're very good at chemistry. Chemistry was invented, hundreds of years ago and developed to a high art, and we use it to grow plants. We put chemical fertilizers on plants. We put insecticides and fungicides on plants. We're very good at managing plants with chemicals. The problem is you put those inputs in, and they're very expensive, and those same inputs, only a part of it goes into the plant or onto the plant. Most of it actually goes into the environment and does damage in the environment. Of course, the outcome of, even though that damage to the environment, but also damage to human health. Okay, the product are crops that are nutrient rich, but actually biologically grown crops are more nutrient dense than crops grown using conventional chemical agriculture. And I'll talk about that a little bit too, as I discuss. Biological agriculture is more recycling. This is actually what plants do in nature. This is how they work in nature. They use microbes, and they use microbes to get nutrients. They absorb those microbes into their tissues. They extract nutrients from those microbes in their tissues. You get less waste, less loss into the environment, almost no waste, and into the environment. It is a much cleaner agriculture, and biological agriculture is much closer to what plants do in nature. This is what they actually do. The agricultural products are cleaner. They're nutrient rich. They tend to be higher in phenolics and antioxidants and other kind of mineral nutrients that go into the. And one estimate is they're 30% richer in nutrients than conventionally grown produce, at least in some cases. So they're better produce, better products from agriculture when they're grown biologically. This is my illustration, not an artist, but I made to the left is an illustration of what we actually do in chemical agriculture. We try to provide nutrients, NPK, right, fertilizers as chemicals. We spray them on plants. And to do that, we try to get them to go into the plant. And what I've illustrated here is a plant cell, a little square thing. And you see, I put a little funnel there and all these NPKs here going in. Most of them don't go in. When we spray fertilizer on plants, it goes into the environment mostly. Little bit, like 30 to 40% will actually go into the plant cells, into the plant tissues themselves. So most of it is lost. And this illustrates that. So this conventional agriculture, chemical agriculture, very inefficient. This is not actually how plants grow. They don't grow. Plants in nature don't require any chemical fertilizer input. They grow using microbes and microbiology. Biological agriculture is closer to how plants grow in nature. And over to the right, this kind of shows what happens in biological agriculture, and that is a plant cell again. And you can see inside the cell, these bacteria, and these are the bacteria that the plant absorbs into their tissues from the soil. That's why you have to have a good, healthy soil with lots of rich microbiology. They absorb them in. And then once the plant has them in to the cell, the plant will extract nutrients from those microbes. And I'll talk about the details. But the point is, this is an endogenous nutrient supply, an extraction, actually, using these bacteria. And yes, the plant gets these from the soil, but then they put them in the cells, and then that's when they extract nutrients from them. And I'll show you how that happens. This system is 100% efficient using biology, using microbes, 100% efficient, or very close to 100% efficient. There's no loss. And it's much less expensive than chemical agriculture. Plants are actually better, hardier when they obtain nutrients using this internal system, this rhizophagy system from endophytes. Okay, so endophyte, this is a term, you can see it up here, endophyte, internal plant. Any microbe that goes into a plant can go into cells, it can go between the cells. This is actually a fungal endophyte. You can see the hypha of the fungus now in the plant. This is in a grass, endophytic fungus. So fungi can be endophytes. Bacteria can be endophytes. This is an endophyte from tomato, actually, this bacterium called Micrococcus. We pulled it out of tomato, and then we put it in another plant called Rumex crispus, or curly dock. The scientific name is Rumex, but it's curly dock. Here in the United States, we call it that. It's a common weed, but we can remove the microbes. Then what we do is we put the bacteria, and we put this Micrococcus on it. On the seed, actually, where we didn't have other microbes, the seed germinated, the root comes out, and then the bacteria colonize the root. And you can see them now inside the root cells. These are the root cells near the root tip, near the meristem, in the meristem of the root of the plant. And you can see the walls out here. You can see the bacteria, these clusters of these bacteria, these tetrads, clusters. And there's nothing like chloroplasts or mitochondria, any kind of cell structures that look like this. They don't look like that. They're single-celled, but you can see these are all clusters. This is our Micrococcus that the plant absorbed into the cells. Where are these bacteria from? These bacteria are all soil microbes. They all come from the soil. They live in the soil, around soil organic matter. This is why we have to have healthy soils. This is why we need to try to build up our soils, increasing the organic matter in the soil, increasing the carbon, increasing the biology, little by little, because the plants have to have those microbes. They need those microbes as endophytes and for rhizophagy cycle to get nutrients. These microbes, once absorbed, also the plant will then vector some of those microbes from the roots into the leaves and into the seeds, onto the seeds, so that then the microbes, the best microbes the plant can get out of soils, the plant puts on the seeds. And that's important because the seeds then will carry the microbiology that's critical with the seed. And so when the seed germinates, the bacteria will go out and colonize that seedling and enable that seedling to grow. And they're extremely important for seedlings. So the seed vector microbes are important. You don't want any sterile seeds because seeds become really very weak when they don't have their microbiology. So here's an example. This is a grass. This is really a fescue grass. And it has on it, the seed itself has on it what's called actually a lemma around here, this dead plant tissue here, leafy-like tissue on the seed. That lemma actually carries microbes. There's a little community of microbes that go on that seed. And we think that the little coverings of seeds, that, for example, the little leafy coverings, these lemmas and paleas that associate with seeds, we think they actually are adaptations in the plant to carry microbes. So this is very important. And when the root comes out, the microbiology comes off the seed and then colonizes that root and colonizes that plant. But also there are bacteria inside that seed around the embryo. So you get bacteria on the surface, you get bacteria inside. And if you do anything, for example, to clean seeds and you try to seed treat them to kill whatever microbes are on the surface, you're going to knock out many of the microbes that are naturally carried on seeds and that are needed by that seedling to grow. So you have to be careful with any kind of sterilization practices. So tomato, here's another example. Tomato. We think of tomato as totally sterile inside. The fruits, it's not true. In fact, the cells in the fruit have bacteria in them. They're inside there. And if you look around the seed, there's lots of bacteria around associated with the surface, and they'll be on the surface when that seed is mature. And there are bacteria that are inside that seed around the embryo and even colonizing that embryo. Okay, so there are bacteria inside and outside the seed. Okay, so even tomato seeds, all seeds carry their endophytes inside and on the surface. So you have to be careful. We have lost endophytes in many of our crops because of how we treat seeds. And so anyways, so be careful with seed treatments. That's my point. Okay, now I'm going to talk about the nutritional process in roots, and that's the process that I referred to as rhizophagy. Rhizophagy, again, root, rhizo, means rhizo meaning root, phagy referring to eating. So this is the root eating process, the process whereby plants eat soil microbes. And this was discovered actually in Australia by a group of investigators in Queensland, the University of Queensland. And what they did actually is they took yeasts and they took bacteria, and they labeled them with green fluorescent protein so they could look and they could tell, what the bacterium was that they labeled. And then they put those bacteria and those yeasts on seedlings of plants, tomato and Arabidopsis, and then they let them go into the seedlings. And what you see here, if you look at E down here, this is actually a figure from their paper, this whole plate. But if you look at E, you can see a root hair, and you see the little green dots. Those actually are yeasts that were labeled, and that the tomato plant or Arabidopsis plant absorbed them into the root cells. And so now you see them in the root hairs. Okay, so these investigators saw this, and they noticed that in time, the bacteria appeared to disappear from the root hairs and from the root cells. And so they said, well, these microbes appear to be degraded by the plant. And so they said, this is actually appears to be a nutritional process. And so they named it, actually a couple of years later, they named it rhizophagy, the root eating process, right? Their paper, they published, this is in 2010, so this is recent. This is why you haven't heard about this long ago, because it's pretty recent. It was published in 2010 and published in a journal called PLOS One, open access. You can get to it online easily. They titled it "Turning the Table plants consume microbes as a source of nutrients, okay, as opposed to microbes consuming plants as a source of nutrients, right? In fact, when we have lots of microbes on plants, lots of bacteria on plants, we tend not to have problems with pathogens as much. And part of that is because the microbes are part of the immune system of the plant. These are symbiotic with the plant. They end up being symbiotic with fungi too, potential pathogens, and they change behavior of the pathogen. But I'll talk about that when it comes. The point is, these symbiosis are very important in this rhizophagy cycle, and the microbes involved with it are very critical for all aspects of the plant. Okay, so it turns out bacteria work, all kinds of bacteria, and this shows this where the bacteria here, the image below, there's bacteria here, you can see below a root hair, and you can see the bacteria inside the root hair, so inside the cell. Okay, yeasts, and you can see here's actually a root hair again, and this is the yeast. These little spherical dots are the yeast inside. Inside, they don't look like yeast because they don't have their cell walls when they're inside the plant cell. But I'll show you, I'll explain what's going on. But they're actually are the yeasts inside the root hair there. And you can see them coming out of the tip of the hair. And this is part of the rhizophagy cycle. You see them coming out. Small ones here, bigger ones as they mature. And actually algae, green algae can be absorbed into plants. And you can see it here. This is a chlorella. And you can see it here in a root hair here. And you can see the alga in there actually losing its chlorophyll once it's been absorbed. So many microbes can be absorbed into plants. We think actually protozoans can go into plants, and I'll show you. The plant is doing this. The plant is absorbing these microbes. These microbes aren't going in on their own power. The plant is taking them in. At least I should say, we don't think they're going in on their own power. We think the plant absorbs them. We can see them going through the walls because at the root tip is where the walls are soft, and that's where they go in, where the plant cell walls are soft. So we don't know the exact process, but it's pretty clear they're going through the wall. And this whole process appears to be under plant control. So most likely it's the plant that's absorbing the bacteria rather than the other way around, the bacteria entering on their own. Okay, so this is a diagram of a root tip. And what you see to the left over here, you can see root hairs, where the root hairs are forming, and to the right here is the tip itself, and there's the meristem here. At this meristem is where the plant secretes exudates, and those are sugars, organic acids, maybe nitrogenous forms, other kinds of nutrients that the plant secretes. But those nutrients that are being secreted are obviously nutrients for the soil microbiology. So the plant is cultivating those microbes, but they are attractants for those microbes. And so the plant secretes the exudates, and then the microbes come running. They come swimming. They have flagella. If they're growing, they grow to where the source of those nutrients are, and they grow right there at the tip. They're attracted to the tip. And then the plant takes them into the cells there at the tip of the root tip, and then extracts nutrients from them. And I'll show you how that happens. Surviving microbes will go into the root hair initials, and then those microbes will produce hormones that cause those root hairs to grow. If we have no microbes in the root hairs, then the root hair initials, then you get no root hair growth. So you have, in order for root hairs to form on a plant, you have to have bacteria inside the plant cell, inside the root cell. Okay, so if there's bacteria there, then the bacteria will trigger growth of the hairs, root hairs, and then as those hairs grow, those bacteria are ejected. Some of the bacteria are ejected out to the soil, where they'll reform their cell walls and they'll go out and acquire more nutrients. They'll become soil ready again, and I added something, reform their cell walls. That's because when they're inside the plant cells, they don't have their cell walls on them, and I'll explain that in more detail. But once they're ejected, they reform their cell walls, they go out and get more nutrients later to be attracted again to these root tips. So that happens until all the bacteria are ejected from the root hairs, and then root hairs don't grow anymore. They have to have the bacteria to grow. So plant growth is really dependent on the presence of these bacteria in plants. Okay, this is a diagram from an Austrian scientist, Dr. Harriet Mella. She's just showing these bacteria being attracted to, absorbed in, releasing their nutrients. Then they end up going into these hairs, and you can see them, the root hairs, trigger root hair develop, and then going out the hairs. Okay, so that little illustration, that just shows it again. You see it going in, attracted, losing nutrients, and then ending up going into the hair again. So that's the process. This is the cycle right there. So this rhizophagy cycle happens at all of the root tips. So if you consider a plant like a grass, like corn, for example, this is a diagram of a corn root. You can see all these roots, but you see these little side branches, all these little things sticking out. Those are all branch roots. At every branch root tip is where rhizophagy cycle is happening. So at all of those root tips, those thousands and thousands of root tips, the plant is getting nutrients from those bacteria in the soil. So you could consider more root tips, more root branching, more nutrients plants are getting from soil microbiology. And in fact, when there's more soil microbiology, you get more root tips and more soil branching. So it's a direct correlation there between those things. This actually is a root tip now. And you can see the root, the meristem is here. You can see the bacteria right here. You can see all those bacteria. And the closer, notice the closer you get to the root, the denser they are. And they're being absorbed in right at that tip. Why are they absorbed in at the root tip? Well, there's exudates there. But the other thing is the cell walls, the plant cell walls are soft at this meristem. And so they can be taken in. So they can basically be squeezed right in, pressed right into those cells, right through the cell walls. And we don't know all the processes, like I said before, but the cell walls are soft, so they go right through those cell walls. Now, once those cell walls become thick and they're in older cells, more mature cells, they can't get out. They're essentially trapped inside those plant cells. So in fact, one role of the plant cell wall may be to trap these microbes in the cells so that the plant can extract nutrients from them. This shows a tip of a root, and it's stained with crystal violet. This is a bacterial stain, and that'll stain the cell walls of the bacteria purple. And so if you look at the tip over here to the right, you can see those little purple dots. And if you look below, this is just an inset. You can see purple dots, beautiful purple dots. The dark purple are the cell walls, okay? And this behind that meristem, so the bacteria enter here, you can see they still have their cell walls. Then you see over here, a little bit older, you can see now they look different. It's no longer spherical. And if you look below, you see it's very angular. What's happening is the cell walls are being oxidized off of the bacteria. The plant in this zone is oxidizing bacteria using superoxide, very potent form of reactive oxygen. And this was the very first digestive molecule that eukaryotes, that plants developed, that plants and higher eukaryotes, essentially, animals and plants and fungi use this superoxide. So it's very potent digestive molecule, and it comes from oxygen. But you can see what it does. It takes, the cell walls are coming off. Look at close-up of this a little bit more. And you can see how irregular those are. Those are the cell walls coming off of the bacteria. And the little pinker things in there, those are the bacterial cells themselves. So the bacteria lose their cell walls, and you have protoplasts then. Here, this shows these bacteria. This is what they look like in their protoplast form inside a plant cell in a root cell. And you can see they're actually growing in here. They're a chain. You can see there's a chain of them. And you can see 1, 2, 3 there where my arrow is. The smallest is blue. That's because it's full of protein. The blue, we put a protein stain, aniline blue, that stains for protein. And so you can see the protein in there. But notice the older ones, the bigger one. They're not blue. The proteins are mostly gone in those older ones. The superoxide will go through the membranes and will continue to extract nutrients from those bacteria. And eventually, some of these bacteria will break down completely, but many of them will survive in the plant. So plants are taking whole communities of microbes from the soil, they're absorbing them, and then they're extracting them for nutrients like this. And And then they're alive. They're actually alive. This is one of my graduate students, Celeste Zhang, but you can see what she did. She actually labeled a bacterium with a fluorescent stain called M. cherry, and then she fed it to a plant. She fed it to, what was this plant? I don't know what this plant is, but it was lettuce or something, whatever it is. But then she looked at it with the confocal microscope, and that's a laser microscope that allows us to look at fluorescence. And so very tiny things with fluorescence microscopy. And you can see here, she scans through these root cap cells. You can see the M. cherry stain, the label, the bacteria, the orange, they're inside. The blue is the cell wall. So you see they're inside there. So we essentially did, it's not novel, but we essentially did what the Australians did with this other bacterium. And so you can see, and now she's scanning through it. And so you can see that. These bacteria are going into the cells. This is a diagram of a root cell. The gray is the cell wall. And the center is the cytoplasm in here. And the bacteria go into this, underneath the cell wall, in this zone around the cell. You can see the bacteria labeled there. And some of them will go into the cytoplasm. And they'll even go near the nuclei. But the plant will process these microbes around and around and around like this in the process of cyclosis. The plant is moving these microbes. The plant is managing these microbes in their cells to get nutrients out of them. So the plant is doing this process. The plant is processing these microbes. And one aspect of that is this cyclosis. And I'll show you that. I'll show you an image of that. But once the microbes are in the plant cell will secrete superoxide. And you can see here, this superoxide here, this 2-, all this is oxygen. It comes from oxygen in the air, and the plant has an enzyme called the NOx enzyme. I'll show you that. They pull oxygen apart, so oxygen has to be in the soil around the roots. They get that oxygen, they pull it apart using this NOx enzyme, and they make 2O- superoxide. And then that's what they use to manage these bacteria, to manage these soil microbes, to extract nutrients out of the soil microbes. So I'm going to go into details with that. Okay, this NOx enzyme, this is in the root cells, and it tends to be in the plasma membrane or in the membranes in general of the root cell. And this is a diagram representation of it. And you can see molecular oxygen has to be soil air. So if you have soil that's flooded, you have no soil air there. Plants cannot do rhizophagy cycle because they cannot make superoxide because they have no oxygen from the air in the soil. So in that case, plant growth is going to be, if it's flooded soil, it's going to be repressed, highly repressed. You're not going to have good growth. Plants are going to be weak because they can't do rhizophagy cycle. They can't get the nutrients they need from plants. They can't get antioxidants. They're not going to have antioxidants. They're going to be really weak and susceptible to disease. And notice also this enzyme has iron as a cofactor. Okay, and that's significant because if we use excessive chelating herbicides like glyphosate, could be a problem because glyphosate chelates iron. And if you put too much glyphosate on soils, chelate the iron, especially around plants, and then plants cannot do rhizophagy cycle because they cannot make superoxide. So they can't extract nutrients from bacteria. So you have to be careful bear in mind that this process is happening. So, and be careful with chelating herbicides.
James, can I jump in a second with a question here?
Yeah,
So
you bet. Sure.
Two questions pertinent to exactly where we are in your presentation. And one of them is from Warren. He said, what's in it for the microbes, the quid pro quo for being abused inside the root? And the second question is a little bit more complex. It's from Paul Swartz. He says, you mentioned not using ionic nitrogen containing seed coatings, which causes a plant to disregard the rhizophagy cycle. Is this the same for ionic nutrient application later in the life cycle? Is a plant's ability to use this process mutually exclusive with never having seen certain ionic nutrients? on to say, to put it differently, if a plant doesn't start its life knowing rhizophagy, can it ever? And if so, what practices can farmers implement to rehabilitate plants towards rhizophagy?
Yes, that's a mouthful. First, I'll do the first one. The first one, what's in it for the microbes, right? So the microbes are being absorbed into the plant. The plant actually is farming these microbes, and it'll take a few microbes in at the root tip, extract nutrients from them, increase their numbers, and then put them back out into the soil. So the plant is actually increasing the microbial numbers of the microbes that work with it, right? And not only that, the plant is selecting microbes, improving the capacity of the microbes to work with it by, for example, by selecting microbes that are not they don't degrade too easily, selecting microbes that will break down and release their nutrients, right? Those are the ones they have to be able to, for example, form these protoplast stages inside in order to function properly. If they don't, then they're not replicated and not put out. So you can see the plant is actually selecting these communities for the ones that work with it, increasing those in the soil. Okay, so that's what's in it for the microbe. But it really is, I mean, it's the same kind of a symbiosis. It is a symbiosis. Same kind of a symbiosis that we have with, say, cows, right? Except that we're eating most of those cows. That's still a kind of a symbiosis. Except that in this case, most of the microbes, or at least the microbes that the plant relies on most, are the ones that it cycles. And so the plant is actually increasing those microbes around the soil. Okay, and the chemistry, yes, there is an issue with using the chemistry and using biology. Typically, if we overuse chemistry, we make it difficult for the biology to work. There is a kind of a mutual exclusivity, but there also is a middle ground. There also is, because we know that plants naturally use microbes, soil microbes and endophytes, right? They use these microbes to get the nutrients that they need, but they still have the capacity to absorb nutrients from the environment. And it may be something, we don't know the exact ratios, and it depends on the plant. If you look at wild plants, it's gonna be a lot different, but plants get some nutrients absorbed from soil water. Okay, and those would be the ionic forms that you're talking about. Not all their nutrients. So if we're over fertilizing with chemical fertilizers ionically, we are going to paralyze the rhizophagy cycle in this process where plants are using to cultivate microbes. They'll just stop doing it. They'll just stop doing it. You'll be able to tell because roots aren't as branched. You'll see fewer microbes around the roots. All kinds of things will start happening. And if you're using exclusively chemical fertilizers. So there is a middle there. And I advocate that middle as opposed to just one or the other. But using chemical fertilizers more like biostimulants. Yeah, go ahead, Sue.
No, I was just going to say, so what we're saying is that the actual plant's full expression is affected when we take away the endophytic biomes.
Yeah, plants don't work the way we think with chemical fertilizer. That's not how they work. Their development, the things that they do depend on microbiology. Their development depends on microbiology. They don't develop properly if you feed them fertilizer. They need these microbes in them. They don't develop the chemistry that they normally would get. They don't the hardiness without it, without the microbiology. So the microbiology has to be there. We weaken plants. We make them weaker when we give them chemical fertilizer. That's not how they work. They don't work like that. In nature, they don't work like that. This is something we invented. We invented that. And we are so brilliant but we're suffering the consequences from our own brilliance.
Yeah.
And in a ways, native communities were growing plants biologically. They were very much growing plants biologically. I mean, for example, the Iroquois Indians used something they called the Iroquois corn medicine. And they would go out and get endophytes from wild plants and bring it in and take their corn. And they would put it in, they'd get their endophytes from the roots of plants, from wild plants, and put it in water. And then they'd germinate their corn in it to get the bacteria to grow onto their corn root. Once it germinates, then they'd go plant that. So they're getting endophytes. They were growing with endophytes. They were doing, native peoples were doing it. The people intuitively were growing with microbiology before we invented chemistry. So anyways, I keep going. So development is screwed up without microbes. I mean, for example, if we take a seed and we remove all the microbes on it and we germinate the seed to get a seedling, the roots that come out don't have root hairs. They have no root hairs. Oftentimes the roots will lose their gravitropic response. Gravitropic response is when the roots grow down, but without microbes, the roots don't grow down. They'll grow in the air, sometimes just stick in the air, or they'll grow on the surface. Rarely will they grow down. We don't understand why that is. We do understand about cell growth and elongation, root hair elongation. That's hormones. We know that. What causes the gravitropic response? With endophyte mediated, we don't know. Also, with endophytes, with microbes, plants show more root branching. They show more shoot and root elongation. So big effects. Plants don't develop properly without microbes. This shows a Bermuda grass where we remove the microbes. You see the root here, root tip there to the left. And you can see the root here, older part. Notice there's no hairs there. You can see some little bumps there. Those are the root hair initials, but there was no bacteria there, so they don't elongate. So these plants they're evolved, they're made, they're designed to have microbes in them. This shows the same Bermuda grass, different seedling, but in the same experiment where we remove the microbes and we put one back on, the Pseudomonas bacterium, we put it back on, and now you see hairs immediately. And we go to you look at older part, you can see these hairs, look at all the brown dots. Those brown dots are the bacteria. We use the reactive oxygen stain to see the bacteria. All the bacteria, those hairs are full of bacteria inside those hairs. And those little dots on the leaves and the roots, on the root cells, those are bacteria inside the root cells. They're everywhere. Here's a close-up of one of the roots, and you can see the little brown dots. Those are the bacteria inside. Those are bacterial protoplasts. So they actually are wall-less. They don't have cell walls there, but they're inside. And there's so many. The plant brings a few in and makes a lot more inside. This is tomato. It shows a similar thing. You can see tomato seed where we remove the bacteria with streptomycin antibiotic, and you see no hairs. Same age, no treatment where we did not remove microbes here, and you can see the root hairs forming right there. That's tomato, just to show a crop. Here's tomato roots. You can see where we remove the microbes. These are root hair initials. So if you look to the right here, where there were no microbes in the root hair, where we sterilized the seeds to remove microbes, they had these root hair initials. You can see this little bump, but there's no bacteria there. Compare that to the left now, we had a root hair initial, and you can see bacteria in there. And then that leads to growth of the hair. And you see them being pressed out here at the tip, and you can see it ultimately, this is older, you see them out at the tip, you see more and more microbes. But what's happening is here, they're at the tip, they're secreting growth hormone, and I'll explain that. And that causes this growth spurts actually, and I'll explain how that happens. It's a really cool process. I show you the details because I want you to know this is not a mysterious process. This is something that we all can see, we all can understand. It's not complicated molecular biology where we're looking at pathways, where it's not complex. This is something all of us, every human can understand. And when we see it, it'll change how we see plants and how they work. This is actually a sedge from one of the a desert island actually in Bonaire in the Dutch Antilles, but it's these limestone outcrops. These little sedge grows in these little pits, and they don't have soil, but they have lots of microbes. This is the root hair below, and above, two of them. The above one is living, and you see the movement there, and below one is not living, but it's stained, and you can see where the bacteria are. You can see where the brown are. Those are the bacteria. They're all in these hairs, and now you can see this is now a living root hair. This is something you've probably never seen before, because if you pull the hair out of the soil, you move this root hair and you break the cytoskeleton, and they have a little skeleton, and that cytoskeleton is needed to make the cyclosis, to make this movement. But these little shadows you see, those are the bacteria going around and around and around. That's how root hairs actually work. That's how they process these microbes. This shows a root hair, one of these sedge root hairs, and you can see there's clusters of bacteria there. They started from probably a single bacterium, right? One bacterium entered, nutrients were extracted, then the plant replicated those microbes, making lots of copies. That's what you see happening. So a few microbes go in, the plant produces many and go out. So they're farming, the plant is farming these microbes. This actually shows a hair. You can see the movement here. This is the cyclosis. You see what's happening. The bacteria are going here. Little by little, they go to the tip. This is where the wall is thin and loose, and the bacteria then accumulate here at the tip. You can see them bumbling around there, bumbling around like little ghosts, bacterial ghosts at the tip. Every now and then one gets caught and pulled back. This is how they work. This is how hairs work. But at the tip, those microbes are secreting en masse. They're secreting hormones. They're secreting ethylene. They're secreting nitrogen. And those, and I say nitric oxide specifically, we count that as a hormone, but nitrate even behaves as a hormone in the extent that it works in very tiny amounts. And what happens is they secrete those substances, and then that causes this hair to grow. When that hair grows, the bacteria that are here at the tip will be ejected out, and then they'll reform their cell walls. The ones that remaining will stay here, and they'll keep circulating around, and they'll increase again, and they'll go back into the tip. Then that'll cause another growth spurt and ejection. And I'll show you, I have a diagram of that. Oh, this shows one of these hairs. See how fat it is? It's full of bacteria. Hairs are really microbial cultivating structures. They're microbiome structures. We think they're about absorbing nutrients, but it looks like they're actually about processing microbes and getting nutrients from them, yes, but processing microbes internally. You can see these microbes outside where they come out of the root hair. This actually shows a diagram of that. I'm not gonna go into the details about what this diagram shows, but basically it shows the growth spurts that happen. Accumulating in the tip, forming hormones, causing an ejection. They go out, they form their cell walls again, they form flagella, they swim out, get more nutrients. These that are on the sides stay in there, they're replicated again, cyclosis, goes to the tip, makes another cluster, and then it causing ejection. So you have these growth spurts, and I'll show you, this actually shows that. This is a root hair, and it has some bacteria in it that were not fully ejected. We got this bacterium from a plant, and it wasn't quite adapted to the, in this case, Bermuda grass, but you can see the clusters, the darker areas where bacteria remained in. Then there was a growth spurt, and you see some bacteria then accumulated there, another growth spurt, and you see bacteria there. A growth spurt happened, but left these inside, right? And then you have some more that accumulated at the tip, caused a growth spurt. Some were left and so forth. So you get this banding, but they are a result of growth spurts that happen with microbes staying in there. This is tomato showing the actual ejection. If you look at 3, you can see there are little pores down there, and you can see their bacteria are getting pressed out of the pores. Because they're inside the root hairs as protoplasts, inside the root cells as protoplasts, they go out of these little tiny holes. They'll squeeze right out. It's like an ejection, like a syringe, right? Ejecting those microbes right back out into the soil. The plant also gives them a little bit of exudate so they can reform their cell walls and their flagella if they have flagella, so they can go swimming out back into the soil. So the plant makes them soil capable. All the evidence we have suggests that this is a nutritional process. It's critical for plant development, no doubt about it, and plant health. But it's a nutritional process, and this actually speaks to the kind of the middle ground using ionic chemical fertilizers and microbiology, because we do know that plants can get some nutrients, many nutrients, all their nutrients they could get dissolved in soil water. That's not how they work. That's not how they function, but they can. And so we know we can use fertilizers to do that. Certain percentage of nutrients do come dissolved in soil water. We also know that plants will associate with mycorrhizae, fungi in the soil to get nutrients. And so certain of their nutrients come from that. But they also do this oxidative extraction using rhizophagy cycle. This is the part we didn't know about before.
James, another question exactly on that topic.
Yeah.
And this from Danny on YouTube. Hi, Danny, and hello to everybody who's with us on YouTube. I can't actually see you, but thank you for joining us. The question is, do we know the relative percentage that rhizophagy plays in plant nutrition compared to other paths, mycorrhizal partners, saprophytic partners, predatory prey cycles of bacteria and protozoa?
Not in detail. We don't know. And I can tell you this, it depends on the plant. Okay, if you have a wild plant, all those percentages are going to be high. But if we have plants where and soils where the microbiology has been damaged, all of that falls, right? So if you go into nature or you go into a very microbial rich, healthy, regenerative, organic soil, you're going to be higher rhizophagy nutrient acquisition than if you go into a conventional agriculture field and try to measure it. But you know, the level that you're talking about, no, we do not know. We do not know the various percentages coming from each source.
And back to what you've mentioned about the Iroquois and how they actually extracted endophytes from wild plants. Can we do that to enhance the endophytic bacteria and plants that we grow in the soils that we have? Can we do it ourselves? And if so, how easy would it be to do that? And are the endophytes species specific? Would that be an issue? I mean, why aren't we squeezing the juice out of cactuses and spraying it all over our plants to make them hardy?
And some people are scraping juice out of cacti, out of yucca and cacti, and putting them onto plants, creating microbial products with them. So some people are doing that. Yeah, we can do that. We do it experimentally in moving whole communities from plant to plant. There is an issue, and that is there may be an adaptation issue, and that is that sometimes you put them in and there's a suppression in growth. It reduces the growth. What has to happen is you put them in, and then you have to go through a selection process where you select for better a combination for that, right? This is a kind of a breeding that has to go with that. But yes, it's easy to do. You can do it. We do it all the time to test for nitrogen fixation by endophytes, and especially with our nitrogen fixing corn project, we're moving those endophytes into different grasses to see what they're doing, and different plants in general. Yeah, so you can do it, and people are doing it, and you can do it. Yes, it works.
How?
Yeah. So, did that get it, Sue?
Yes, you did get it. My next question is, if it's so easy, how would any of us here that were thinking about trying to do this, how easy would it be? Yeah,
so what you can do is, and this is kind of what this is the process, and I didn't go into the details, all the details of what the Iroquois were doing. They would take their roots, grind them in water, then they would warm it up, not much, not necessarily, not long enough to kill all the microbes, but long enough to reduce the microbial titer. And at the same time, they would wake up the endospores. So the gram-positive endospore formers that are in there, wake those up. So a lot of these root bacteria actually gram-positive endospore formers. So the heating, so they would be like 50 degrees centigrade for maybe 10 minutes, something like that. And that reduces a lot of the swimming microbes. And then they would take that, cool that down, and germinate their seeds in it. So you could do that. And that way you get a subpopulation of microbes, a subcommunity from whatever were in those wild plants. That's the way to go. That's the way where you reduce any inhibition, because sometimes if you have too many microbes, too much diversity, you're going to get reduced growth. That's where if you put so many microbes in there, you have to then kind of select to try to balance the system again, right, with selection like that. Yeah, it really is quite easy, though. You just have to play with it. This is where you have to be experimenters, and you got to be willing to stick with it and do a couple of seasons where you're playing with it. You could change your crops. You could make your crops more resilient, more resistant by adding this microbiology to it. You could just by growing them in healthy soils. This is where if you keep your seeds, right? I know that there are big companies that are wanting to control the seed supply, but you have to fight that, because you have to have control of your seeds. You have to be able to grow those seeds in soils that are rich in microbes, or at least soils repeatedly, grow them over and over in those soils to make them so that they have the right microbes to grow in those soils. So yeah, and I'll talk a little bit about that. Sue, did you want to ask something else before I move?
No, that was all.
Okay. So why do these microbes affect plant growth? It's because they produce hormones. They're producing ethylene and nitric oxide, and nitrate acts like a hormone. And why do I say that? It's because it's produced in very small concentrations inside plant cells around these microbes, the nitrogen. And it just makes the growth of that cell just a little bit, kind of like hormones, affecting like hormones, even though it's sourced from a microbe inside those cells. There is this process called habitat adaptive symbiosis. This was described by scientists Rusty Rodriguez and Regina Redmond and some other authors. But this is an idea that plants growing in extreme habitats or stressful habitats will absorb the microbes that they need in order to grow and thrive in that environment. And that's the habitat adaptive symbiosis. That is, the plant themselves will make the symbiotic association to enable them to grow in that environment. So that's why you need to keep your seeds, because the stresses in your soils, you will cause the plant to absorb the microbes from those soils that enable it to grow and thrive in those soils. And that may take several generations as you do that. So this habitat adaptive symbiosis is very important. The microbes cause epigenetic change and even possibly genetic changes in the crop. So microbes actually will increase the capacity of the plants to adapt. And part of the reason for that is because the microbes actually regulate or control plant development. They control development. They control whether plants become oxidatively stress tolerant, whether plants are more resistant to oxidative stress or salt stress or whatever it is. So they alter development of expression of genes in the plant. They'll produce ethylene and nitrate inside the plant, and these act as what we call nucleomodulants, or they'll affect really the DNA expression in the plant. And that's the point. They'll modulate plant DNA. And there's a process called endoreduplication that they stimulate, that at least the nitrate and ethylene will stimulate, which means basically division in the plant nuclei. And when the plant nucleus divides, then you get things like jumping genes, transposons, things like that can be expressed. So you see the differential expression happening. Okay. This is really emphasized because these endophytic microbes, some of them go into the nuclei. They become intimately associated with plant DNA and affect DNA expression. This is a potato root cell, and you can see the cell here. You can see there's the kind of blue areas where the bacteria are around it. These are bacteria inside that root cell. This is the nucleus, and you see these little bacteria. Those are those little dots, donut-shaped things. Those are bacteria around that nucleus. They go actually into the nucleus, and they'll express, and we think it cause nucleomodulins. They'll change the expression in the DNA. This shows another plant. This happens in all plants. This hosta. This is a flower, right, that we have here, but you can see the epidermal cell here. You can see the nucleus. You see the bacteria coming out of the nucleus. They're in the nucleus. They're in the nucleus. Here too, hosta again. You can see the bacteria coming out, the nucleus. Okay, this is corn. This is one of our land races. This is an American nitrogen-fixing corn. This one is from Mexico, but this is the pollen of the corn. Those bacteria actually go into the pollen grains and the pollen mother cells, and that's where, you know, pollen is being produced. So the DNA, you're getting divisions going on there. And so we think that the bacteria there are affecting the DNA going into the pollen. And you can see, actually, here's the pollen grain. You see the black area, those little dots, those are the bacteria in the pollen. Well, the same thing is happening in the ovule. The bacteria going through the ovule, going through the, so all parts of this plant are being affected by the bacteria. When you compare crops grown biologically compared to those grown conventionally with chemical fertilizer, right? If you look at the chemical fertilizer crops, you kind of have this situation above. You have uniformity of product. You know, you put these fertilizers out, and that causes these plants to grow, to just grow. You put enough there, and you're going to get big plants. Yes, they're all going to be very similar because, you know, that's how much. And of course, you're going to have all the environmental degradation from issues from fertilizer going in the environment. Biologically, you have plants develop what I call non-uniformity. They're different. Why are they different? They're different because they have different microbes in them. They have similar microbes, but they have different amounts. They have different proportions. You may have, you know, one species that the other one doesn't have. So you have this variability that happens, and so you get variable non-uniformity in plants. Well, that also is your that's also your crop and your food security, because all of these variable plants have different properties. Some are more resistant to stress. Some are going to grow faster under stress conditions. Some are going to have the right microbes that are exactly what you need when there is this drought or there's too much salt or whatever happens. This actually is the way we want to grow plants for the maximum food security. It's going to be a lot more work biologically growing plants because it requires more work than just squirting chemicals on plants. But the benefit, the food supply is going to be safer. Okay, nitrogen fixation happens. I know. Sue, can I have a little bit more time just to cover
Of course
some?
you can. Please do go ahead.
Okay, I want to talk about nitrogen fixation just a little bit. I do see a hand hopped up. Joffre Bell group, I see. Yeah, did you want to ask a question? Did they write something, Sue?
I don't see a question from him.
Okay, no problem then. We'll just keep going. Okay, nitrogen fixation. Okay. All plants fix nitrogen in. We think only legumes do it in root nodules. That's not true. They all do. All plants do. And they do it using endophytes. And more wild plants than our cultivateds, because we've lost many of them in our cultivateds, but they use endophytes. And if you look at the earliest plants, you know, you look at like mosses. We have a group at Rutgers working on these. This is our group here. These are mosses. These are the earliest plants, the simplest of the plants. You see these little brown filaments? Those are little filaments are actually full of nitrogen fixing bacteria. And I'll show you. This is their earliest plant, right? This is the earliest ones. These are pre-crop. These don't even have seeds. Okay. But if you look at one of those filaments, you see these little dots there, the blue around them. That's where the bacteria are. The blue is nitrogen. We stained using histochemical staining. So the purple or the blue is nitrogen that they're secreting around those bacteria. So plants have been doing this. They've been getting their nutrients. Here's one. This is actually a liverwort, another primitive plant. Here you can see the filament, and you can see the bacteria in it, the little blue dots. Those are in there fixing nitrogen. And you see the green part with the chloroplasts. And you see they're green. You don't see bacteria in there. You see chloroplasts. Well, part of the problem is you can't fix nitrogen if there's oxygen, too much oxygen. And where these chloroplasts are, there's too much oxygen. So hence they have these little filaments for putting bacteria in and fixing nitrogen. There's low oxygen in those little filaments. So you see how plants did this. They made little structures where they're going to fix nitrogen. So that's what plants did. Those are the primitive. The process of nitrogen fixation is just this. You know, microbes get nitrogen from the air into, they'll take that and they'll fix it into nitrogen, ammonia, for example, or reduced forms of nitrogen, and then they'll oxidize that with superoxide. Again, that like in the rhizophagy cycle, right? They take that superoxide from the air, they zap it, they hit their, in this case, their ammonia with it, and you end up with nitrate, and then plant gets that nitrate. So this is what's happening. At least this is the best evidence we have of what's happening inside the plant based on our histochemical experiments and our other experiments. And so we have this exchange, this nutrient exchange, and I'm not going to go into the details of that to dwell on this, but it's a complicated process. And I mean, for example, this is data from one of my students, Ivelisse Irizarry, graduated, now working as a professor in Puerto Rico. But she took cotton, put her bacterium on it, and what she showed is when she did that, put the bacterium on it, she got increased expression in this microarray study of nitrate metabolic genes, nitrate assimilation genes, cellular nitrogen compound metabolic process. Again, an indication that nitrogen fixation is happening when you put the microbes in the plant. We've been working closely with Walter Goldstein in the Madaman Institute. He's a plant breeder on corn. And Walter has been trying to breed organic corn. So we've been helping him now for a few years now to breed this nitrogen fixing corn. And what you see here in this image, this is Walter Goldstein here at the Madaman Institute in Wisconsin. What you see here is some of his corn that it comes from these Mexican cultivars, these land races, right, where they're nitrogen fixing. And then you see conventional corn. You can see how much darker this nitrogen fixing corn is. And what Walter has been doing, what we've been trying to help him do, is to show this is happening and to better select and produce corn. And he has already been giving, believe it or not, we have been giving out corn to NGOs in Africa now to start developing some of these nitrogen fixing corn to spread out in Africa. So several, we're active in that process. Walter is more active in it than I am. I'm mostly like a front person, but Walter does all this other stuff. I mostly am educational, put it that way. But in this nitrogen fixing corn, these microbes are all over the plant. They're in the roots, they're in the leaves, they're in the fruits, they're in the pollen. They go all over. They're fixing nitrogen all over the plant. All over the plant.
So James, what you're saying is that the maize plant is nitrogen fixing not only in its roots, but also in its leaves.
It is nitrogen fixing all over it, all over the structure. Yes, that's what I'm saying.
And that makes so much sense given that the fact that there's so much nitrogen in the atmosphere, right? Do all plants do that? All their leaves?
Yeah, all plants do that. All plants do that. Typically, it is the non-photosynthetic cells of plants, like the epidermal cells and the trichomes. Here's, for example, a cucurbit, a calabaza, we call it, but a cucurbitaceae, right? Cucumber family. If you look at you got hairs all over the plant. Okay, these come from the epidermis, right? If you look closely at those hairs, okay, we see they're actually full of bacteria. This is stained for nitrogen. You see the purple, right there. They're full of bacteria. They're making nitrate. There too, you can see the bacteria in those hairs. You can see all the purple. That's nitrogen in those hairs. They also do this in, for example, corn. The warm season grasses will fix nitrogen. They'll accumulate microbes in the bundle sheath in the leaves, in the leaf blades, in the bundle sheath parenchyma, right around the vascular tissues. They'll fill those with microbes and they'll fix nitrogen there. And that's the ideal situation because the bundle sheath is a low oxygen zone. But there, you can see the trichomes, you can see more. And yeah, this is more trichomes. You see, this is sulfur nitrate. This is on hops. You know, if you like beer, hops, the glandular trichomes of hops fix nitrogen. They're full of microbes and they fix nitrogen. Hemp, if you do hemp, cannabis, the glandular trichomes in hemp are loaded with bacteria and they're producing nitrogen. And this is actually these microbes in general now, besides producing nutrients, they're critical for the immune system of the plant. And just very fast, I'm going to talk about this in a couple minutes. Describe one experiment that demonstrates this. This is actually where we grew basil seeds, the little herb basil, right? And so if you look at the left picture, this is the basil seedlings with endophytes, they're non-sterilized, so they have all their endophytes with them and they grow. You see, they look good. Now, basil seedlings with microbes, with all their endophytes in them, and then we add a disease agent, fusarium, which is a fungus, right? Cause damping off, it'll kill them. And you see, there really is not much difference between, you know, so long as they have the microbes, that fusarium does no damage. Okay, but if you look below, now this is basil seedlings without microbes. You see, when we remove the microbes, they don't quite grow as good as they do above. So we've reduced their growth and we know they don't develop properly, even though there's no pathogen here. But then we put the pathogen in there, the fusarium, and you can see now those seedlings are dead. They're gone. They're gone because their immune system depended on the presence of those bacteria. They are sitting ducks without the microbes there. So this is where we sterilize our seeds. We remove microbes. We're making them vulnerable. That's why sometimes people remove their microbes from their seeds. They sterilize them, they remove seed coats, and then they put on fungicides and things like that. That's because disease is going to knock them out without those microbes there. The microbes have to be there. We think we can grow plants exclusively with microbiology if we do it right. We don't need actually all the fungicides and the fertilizers and so forth. We think we can do it 100% with microbiology.
Wow.
Yeah. And of course, you have to do it. That's the trick, right? I mean, actually doing it is different than thinking we can do it. But we think what happens is when you put the microbes in the soil around the plant or in the plant and they go out into the soil, they'll colonize fungi, potentially pathogenic fungi. And many of these fungi, like fusarium, they have two modes of existence. They have a saprotrophic mode where they'll grow on dead material in the soil and they'll just grow slowly and survive and reproduce out there. And then they have this other plant disease agent phase. And what we think is happening is when there's not microbes there to establish the symbiosis with the plants where they go the saprophytic mode of existence, they don't have those microbes there. Instead, they resort to getting their nutrients by attacking plants and they become plant pathogens and you have plant disease. Okay, so these microbes are part of really disease suppressive soils. And so healthy soils and healthy plants with these microbes in them are going to suppress disease. This kind of shows this in culture. And you see bacteria in the middle, and you see bacteria here too, but these are two different bacteria. And you can see the fusarium here. You can see the fusarium just kind of grew all over this bacterium. Here you see the other condition where this bacterium, you can see the bacterium kind of grew all over the fusarium. And it's a lot of white mycelium to the right and less there. It's affected the behavior of the fungus. Here, this shows it. This shows it. You can see to the right without bacteria, you see a fungus there. You see the white mycelium everywhere, all over the soil. Look to the left. This is where the bacteria were there, and the fungus was there, and you don't see all the fungi. The fungi and the seedlings are healthy in that situation. Whereas to the right, they actually are accumulating this fusarium and colonizing them. Okay, this is what happens to the fungi. Here's a fungus hypha, and you can see the bacteria all around it. It's a symbiotic association. That's what's happening in the soil. This is what's happening on the plant. Bacteria going all around that fungus, changing its behavior, giving nutrients to the fungus, making it non-pathogenic. Now, that doesn't happen with all bacteria. I mean, all fungi, all disease agents. Some of them are like rusts, really are specialists on plants. And so the microbes tend to not have as much of an effect. But many of our pathogens have this dual mode. They're facultative pathogens, and they live also as saprophytes. So using microbes, you can favor the saprophytic phase. So you can control disease. These bacteria go inside the plant, inside the fungus. You see these little yellow dots or green dots? Those are bacteria inside the hyphae of the fungus. We can show them inside. They go in, they change the behavior of the fungus. Here, this shows them. You can see them coming out of the hyphae, these little dots coming out. They actually see these bacteria here. They're coming out of that hyphae, believe it or not. This is stained with the fluorescent stain. This is another kind of fungus. You see the hypha is full. This is a zygomycete, soil fungus. You see the bacteria coming out of it. They're loaded. Fungi are loaded. There is this interaction between the plant that has its symbiotic microbes, and then you have fungi, potential agents with symbiotic microbes. The microbes can go between the two, modulating the associations. Now, it may not be the exact same microbe, but you have communities of microbes that, and we do know that the same endophyte of a plant can also go into a fungus and alter the behavior of that fungus. So it's okay. Take a mushroom, for example. They're not free of microbes. I mean, we think of these, they're not pathogenic. No, but they still use bacteria inside to get nutrients. And if you look at the tissue, this is one called reishi, the reishi mushroom. People use it medicinally, but if you look at the mycelium in the reishi, you see hyphae like this. And the hyphae, you can see these little black areas here. Those are bacteria coming out, and you can see they're inside the hyphae. All those are all bacteria. And they're stained for nitrate. The purple is nitrogen that's being produced. There's a phenomenon with wood decay. Wood doesn't have very much nitrogen in it, but as these wood decay fungi work on it, the nitrogen content, and it begins to decay, the nitrogen content increases. That's because these fungi have nitrogen fixing microbes in them. And so that I'm not gonna. So really, I'm going to use this as my final slide before we do some final questions. But with these microbial processes in plants, the rhizophagy cycle, plants get nutrients. Critical that they get nutrients. But the other thing that happens, because plants are interacting with these microbes using reactive oxygen, plants upregulate their oxidative stress genes. They become very hardy. They get more phenolics in them, which are antioxidants. They'll get enzymatic antioxidants in them. They'll become resistant to oxidative stress. And all stress, environmental stress, is oxidative stress. So they become more resistant to stresses. That's climate resilience. That's our food security there, having that. Any perturbance, any climate changes, they're better capable to deal with it. And then they have diseases. They'll change behavior of pathogens, and they'll reduce virulence of pathogenic fungi. So the microbes are plant microbes, soil microbiology, extremely important for crops. And these are the points that I've tried to make in this presentation, and just kind of as a summary, growing plants with microbiology is efficient, much more efficient than conventional chemical agriculture. Seed microbiology is important. We have to be careful with our seeds. Soil health is critical. More microbes, the more the organic material is there, the more microbiology can survive there, the better the plants are going to be. Endophytes are critical for crop resilience. We have to have, and this is why we have to keep control of seeds, grow them on lands, create microbial rich cultivars that are going to be adapted to your land. Bacteria protect crops from disease. It's very critical, very important. This is Jeff Lowenfels. He actually wrote a book about this process called Teaming with Bacteria, The Organic Gardener's Guide to Endophytic Bacteria and the Rhizophagy Cycle. It's been out about a year now. It's only about $25, but he's a great writer, and these are students that contributed to this work. A lot of them did a lot here, and I'm open for questions now. And I'm sorry I went so long, but didn't quite do 90, so we still have some questions that we can do.
Wonderful. And there's the book.
There it is. There it is.
Great book for any of you that don't have it. Do get it. You can get it on Amazon. Really, really, really great book. James, forget about the time. What you've just been telling us is just so mind-blowing, and there are quite a few questions in here. I'm going to start with the ones that I've got listed and see how, if we can get ourselves through this quickly. So Maureen Miner says, sometimes we farmers treat our seeds by fermentation methods. Is this good, or are we destroying the microbes?
Yeah, the ferments, a lot of people use ferments. They're really good. A good way to add microbes to your seeds, to your seedlings. So no, that's people do that all the time, and it is effective. And the other thing that they do are compost washes, for example, and like the bioreactor, Johnson-Su bioreactor, for example, those kind of methods, and all of those methods, I'll add even the Korean natural farming methods, right, where they make kind of a homogenates, where you get microbes out of plant tissue, and then they put those on, and you get an effect. It's a way to move microbes to plants, so you're not damaging plants by adding those microbes to plants.
What's so funny about that is, in my years of organic farming, before I started learning about the soil microbiology, some of the things that we were advised to do, and it's been advised in organic and also in conservation agriculture, sustainable agriculture, is part of this making teas with plant materials. With the outcome expected to be the nutrients that are being extracted from the plant, but we've been inadvertently actually putting a lot of biology back out there without really knowing it. So it's interesting now to make those connections between what's happening with the endophytic bacteria when we were doing those things in the past, where we thought we were just extracting nutrients from the plant as opposed to biology.
Yeah, I mean, this is a theme that goes on. A lot of what we see, plants responding to, and plant anyways, is because of the hidden partner inside plants, right? So this is this invisible that we haven't been able to see. Okay, like your teas, we didn't know they were there, but yeah, that's probably how they're working mostly, is through the microbiology, probably. But of course, those nutrients also add significantly to the plant, but you're adding the microbes there, and that to me, that's the biggest benefit there, adding those microbes.
Absolutely, but coming back to the issue of nutrients, you've mentioned that nitrogen and ethylene act as hormones in the plants, and nitrogen and ethylene, they're produced by the microbes. So what happens with excess nitrogen, chemical nitrogen put on in that aspect as well, but when we're considering it from a hormonal perspective?
It shuts it right down. It shuts everything down. The plants don't do it. If you give them chemical fertilizer, if you give them nitrogen, they just take that, and what the nitrogen does when they take it is it will there's a feedback to the bacteria, shutting down their nitrogen fixation process. So the whole process, the rhizophagy process, at least the process to get nitrogen, when you give it nitrogen, stops. So the microbes don't fix nitrogen, plants don't get benefit from them, the microbes just don't help, they may or may not be I mean, cells get big, yes, they'll grow because of the processes, because that triggers growth, but they're all the other nutrients, like for example, the micronutrients that plants get out of these microbes, doesn't happen, right? So in fact, what we do when we fertilize is we essentially take control of this natural process where Mother Nature is in control, right? Where plants are in control of this rhizophagy cycle process and nitrogen fixing process. We put ourselves in place of the plant, trying to push the plant. First, we're pushing the plant, but we're trying to anticipate all the needs that the plant has and trying to put it in there. And so now we're more and more sophisticated, measuring, do they lack this nutrient? Do they lack that nutrient? It becomes, but that's because we take the place of the fertilizer, instead of the plant's natural process, the rhizophagy cycle, nitrogen fixation by endophytes into tissues. So did I address that pretty good? Okay.
Yes, very good. Moving on, how does seed treatment with chemicals affect the symbiotic relationship with these plants, especially against root rot, sudden death by fusarium, and phytophthora?
Well, it's not a good idea to remove the microbes from the seed. So seed treatments that, for example, increase microbiology, for example, you put some nutrients there, some amino acids or something that might give a little bump to or other nutrients that you might put there, give a little bump to the microbes. That's a good idea. That's a good idea. That's a good kind of seed treatment, right? Because you're growing plants using microbiology. The chemical fungicide seed treatments, that's a kind of seed treatment because we're forced into it, because we remove the soil microbiology, and the soil doesn't have suppressive character, but the plant also is lacking bacteria that would suppress if it's a fungal pathogen, suppress the fungal. And it depends on the situation. I mean, if it is for example, a disease caused by an oomycete, a water mold, right? Because the soil is flooded, that might be a little different than if it's just a fusarium that starts coming in. So it depends on the situation. But in general, you don't want to sterilize seeds. You don't want to put toxins on seeds if you can get away with it. You do want to put microbiology on seeds. You do want to put nutrients that feed microbes. You just want to be careful how much you put on, right? So everything that you do, you have to kind of do an experiment before you do it big scale. Growers have to be experimenters with biological agriculture. It's just critical. That's the thing you have to do.
Absolutely. And I'll come back to large-scale continental experiments after that, but related to that. But whilst on the topic of seed, when we think about those that govern seed and our laws around seed and our standards around seeds, they all seem to be counterproductive to climate smart ag at the end of the day, because seed, you can't buy seed unless it's treated. You can't trade farmer seed. Most seed is going to come in treated with fungicides, etc. Even if you want to bring in organic seed and grow organic seed, for example, in Kenya, you have to do it under absolutely confined. It's funny. We do it under almost like a confined environment for fear of contamination with what would essentially be the right biology for the right seed.
Yeah, a lot of what's happening now with seeds for the past few decades is, with genetic manipulation coming into the picture, and people trying to improve crops genetically, and even before, just cultivars that are specialized, right? Certain properties of certain cultivars. Companies have tried to have patented them, patents, and try to control that. and that has a negative impact on agriculture in that, if they control, if big companies control seeds, they're going to grow them out west, they're going to grow them somewhere, they're going to grow them chemically, most likely, they're not going to have good microbiology. And you're going to take those seeds, they're not going to be adapted to soil. If there's issues in the environment, they won't grow well. They'll also be more susceptible to disease. So what's necessary is that really, and this is what ancient humans have done, is, I mean indigenous peoples, right? Indigenous peoples. They grow their plants on their soils repeatedly, gather their seeds, they're growing their own stuff. They're gathering their own microbiology that adapts that plant to that environment. So yeah, that's what needs to happen. That's why Walter at the Mandamin Institute, that's why we are giving out those corn varieties to GMOs all over the place. It's because we think that with corn, we think that people, once they get this kind of material, they can then start crossing it with their own land races and their material, and then produce cultivars that we can grow everywhere that'll be more resistant than many of our commercial cultivars. So that's the goal.
Yeah. On that angle where you mentioned GMOs, there's a question here from Jackton. He said, I'm working on GMO maize right now. I'm doing the 16s or 16S sequencing. What do I expect on the behavior of the microbes? Do you think GMO crops alter the rhizospheric microbes?
The GMOs do. They have microbes, but those microbes are not as good. That is, the plants don't benefit from them like these landrace corn does. This highly microbiological plants, right? You could take these commercial cultivars, by the way, grow them in soils that are rich in microbes, and in a few years you can get them to recover. So you grow them a certain way, you can get them better. But they do have microbes, but they're not good microbes. They've lost many of the good ones. I mean, you could imagine, because they're GMOs, they're not grown organically. They're not grown under biodynamic conditions, right, with lots of microbes around and organic material and cover crops and so forth. They're not grown that way. So they lost many of those microbes. They're grown with chemicals for the most part. And so they lose that microbiology. They're not designed for biological agriculture. They're designed for chemical agriculture.
Yeah. Yeah.
Many of the microbes have been knocked out. They still have microbes, okay, but they're not, the good microbes are gone.
Well, I'm hoping that so many of our viewers who are here today with us, farmers, are really listening keenly. Hang on to your seed, grow your seed. Don't be fooled. There's a question here from a Dr. Mbaka. He says, with the hot climates in the tropics, the habit of removing all vegetation in the preparation of the land for the next season, leaving the farm naked and under such heat, that's hard to walk barefoot on. It must have serious impact on the soil microbes that the crops will need when it rains. What's your opinion on this, Professor White?
Yes, absolutely. Soils should, if at all possible, well, for one thing, should be covered all the time so they don't dry out. That's one thing. Soil covering, right? This is kind of the principle of regenerative agriculture, soil covering. The other thing is cover crops. Cover crops are extremely important because cover crops, especially multi-species cover crops. And the reason for that is that each plant, each cover crop, you might have 11 or 12 different species of cover crops, but each of those cover crops has different endophytes in it. And they will each contribute to the soil different endophytes that your crop, say, for example, if you're growing corn, that your corn can then take advantage of, can attract and then use. So the more cover crops you have, the more the microbial community, the crop itself, corn, for example, is going to have. Now, these highly microbial rich plants, I should say, they also suppress disease, like this nitrogen fixing corn the landrace corn that Walter Goldstein is breeding at the Mandamin Institute, that corn has microbes in it that will go out and will cause weeds to grow less or not grow at all. We actually have a phenomenon for that. We call that endobiome interference, and that is that the endophytes in one plant that can go out into another plant reduce their growth, reduce its growth. Okay, causing, what that means is you have less weed competition when you have more microbes in the plant, more microbiology in the plant. So these land races especially show that property, and it's due to the fact that the microbes that promote the growth of the corn reduce the growth of some other weeds that compete with it.
Brilliant. It would be really great to get Walter Goldstein's corn.
Yeah.
Find out
Yeah.
where it is that he's distributing on.
Well, he told me already, he told me this morning that there are non-GMO, there are GMOs, non-GMOs in already who have some seed now. Malawi, Tanzania, Zambia, Zimbabwe, and Egypt. Those are the places. Who it is exactly? I know a couple of the people, but you know, I mean, I'm not up on everything, so I can't tell you any more details. You'd have to actually talk to Walter for any details about that. I can just tell you this, that the goal apparently, and I came on to help Walter, but apparently the actual goal is to produce these cultivars and distribute them throughout the world. Distribute them beginning in Africa, I think, is the goal to do this. And that's what we appear to be doing. You know, I'm just kind of watching what we're doing and going along with it and trying to explain how this works and so forth. But Walter, and there's actually an American Indian, Unchatwe, this is Unchatwa, who's in the kind of our three collaborators here, four collaborators, actually, another person involved trying to do this. But that's the goal, is to spread these microbial-rich cultivars of corn as far and wide as we can.
Very cool. So hopefully, I know it's really hard to get you on email, but hopefully you'll drop me an email with Walter's contact so that we can reach out and find out and tap into what they are already are on the continent, so that you can start.
Yeah, no problem. I'll send you the email. I'll send you his email.
Cool.
Yeah.
So 3 last questions. One is, you did mention algae, and some of the endophytes are not just bacteria. Somebody did ask, what role do flagellates and other protozoa play in the process? I suppose that's a rhizophagy process, if any, or is it purely bacterial?
No, we think they eat them. We think they degrade them fully. There, I didn't show the data, but we did some data comparing conventional cultivars to our Walter's nitrogen fixing landrace cultivars. And what we found is that they have, that the nitrogen fixing cultivars are high in protozoans, at least fatty acid. It was a fatty acid analysis. So fatty acids from protozoans, high in rhizobia bacteria that like we see in legumes, right? Nodules and legumes. So they're higher than conventional cultivars in rhizobia, protozoa, fungi, even fungi in there. But again, the fungal behavior changes, right? Because fungi become endophytes as opposed to pathogens. So the other thing, more bacteria, gram-negative bacteria. So their whole communities go into these land races. So that's what happens when you grow them in repeatedly year after year in the same soils, and you take care of those soils and try to minimize the fertilizers. You build up your communities and your plants and your crops, and you build up their resistance and resilience.
And of course, that means that you're building up your own nutrition as well. You're building up your own health, and anything else is health that eats those crops or those plants or
and that is, and I don't always talk about that, but of course, the biological crops are healthy for people, right? They not only don't have the toxins in them that could happen when we use chemical agriculture. at least not the chemical toxins that we put on the plant. But the other thing is they're richer in nutrients. They're richer in antioxidants, for example, phenolics, for example, which are healthier for people. And so they, in addition to the nutrients like manganese and antibiotic antioxidant amino acids like methionine and lysine, those kind of which you don't have in many other conventional crops. So biological agriculture, biological crops are actually better for human health in a lot of ways.
Yeah, I recently attended a national meeting, and actually they were looking at a continental meeting around the discussions were around how to get health, crop health, and nutritional health information out to farmers through extension. And the reason they were doing this, and they had brought in medical experts and hospitals and all sorts of other stakeholders, was because they said that the highest diseases that are not non-communicable diseases are actually coming from poor nutrition. That's what they're beginning to see in hospitals. They're diseases related and illnesses related to poor nutrition.
And this is because of the way that we grow our crops using chemistry. They're not as rich, they're not as healthful for us. And it's also because of the way we process foods. We purify out the carbohydrate, which is still another, we're human, we're brilliant. We're so brilliant. We discovered that we could do that, but as it turns out, we create poison for ourselves. We create poisons. We turn our foods into poisons by doing that. I mean, poisons, there may be a slow poison, if it's sugars and carbohydrates, and that's what we're getting mostly, then eventually when we get older, we're going to get diabetes. That's because we poisoned ourselves with this nutrient that we extracted from plants. If we had just eaten the plant, the way it was, we wouldn't have that problem. And if we grow them biologically, we wouldn't have the chemical residues, and we wouldn't have the problems in the environment with their environmental degradation. So
absolutely. And finally, to the end, we've come to the end of the session. There is one question here, but I did mention, the experiment, and I asked you early on about big experiments. In one of your webinars with John Kemp, was the webinar number 99, and you mentioned that a lot of the work that you're doing is in the lab. You don't do a lot of field work, but there is a lack of data, enough data to support the regenerative movement going forward. And in the back of my brain, I was thinking, well, what if we use all the regenerative networks that we have in the regen producers to conduct some huge experiments where the protocols are put together by individuals like yourself or others, maybe some of your team who are trying to do experiments, but where the protocols are simple enough for us to follow them verbatim on the ground, give you the results in certain ways so that you can actually do experiments at scale using us as collaborators, as citizen scientists.
Yeah, that's a good idea. And that's possible down the road. Of course, part of the issue is they actually make me do a lot of work around here. They have a full-time job teaching and stuff like that. All the lab full of graduate students. It's hard for me to dedicate much time, but I have done this. Like Eric Berg is doing he's one of these internet doctors, and he's been exploring growing plants with microbes. So I've been advising them, and they've been setting up experiments there. So I mean, the same sorts of things. I'm good for collaborating. We can collaborate, but just I have to be careful not to be overwhelmed. It's easy for me to get overwhelmed. I'm also a pretty old guy. I'm 65 already, so I only have so much energy, and I can it's easy to overwhelm. When you're 65 years old, you can only handle so much stress, so it's easy to get overwhelmed. You get it, Sue? You get it? Yeah.
Totally. For everybody in this webinar, you'd be I hadn't actually spoken to James White until this afternoon when he appeared miraculously in our sound check, because I was overwhelming him, I think, with emails. And one of his emails, he said to me, I'll tell you the truth of it, James. You said, sorry, I was like, James, I'm panicking, I'm freaking out. Are you getting my messages? Do you get the link? And you were like, Sue, I'm here. I'm just not married to my email. I got it. And I was like, dare I ask you again if you're going to come to the meeting, or do I just wait and see? And there you were.
Well, it's very important for me to come. It's very important for me to come.
Thank you. Thank you very much. I think that, you know, on behalf of everyone here, by the way, there are people saying you're a spring chicken. 65, now I know why I think the 65 is just around the corner for me. So, I mean, yeah.
That was nice.
Yeah. Thank you from the bottom of our hearts, from Team ICAO, from everybody out there. And we'd really like to thank you very much for joining us, for giving us such a wonderful trip through the realities and, you know, underpinning what we're doing with some really great science and helping us you know, honestly join the dots, and yeah, and you've given us a lot of inspiration going forward with the work that we were doing. So on that note, I'd like to say thank you to you again, and Wes, thank you to Wes. If we can put a spotlight on Wes, thank you for doing the curation of the questions and making sure they got here. I mean, with all the tech that's going on, it's hard to multitask around this. To all of our viewers here, thank you so much. We'll let you know when the recording is up. I think it's live, so it's going to be up anyway now, and you can share that with your friends. James, I don't know how we can, you know, how we can reward you in any way other than to say thank you from the bottom of our hearts. We're going to get in touch with Walter through you, and we're going to spread the seed around. So, yeah, I hope
I can also send the PDF of these slides if you want, Sue, so you could distribute it to anyone that wants it.
Super cool. That would be fantastic. And I'm sure there are lots of people that would love it.
Okay, that's what I'll do. I'll email it to you as soon as we're done.
Super cool. Great. Thank you and good night, everybody. And good night. And thank you, Team ICAO. And see you all next time. Bye-bye.
Bye-bye.